Okie dokie, everybody. We're going to go ahead and get into 1.3 Introduction to Biological Macromolecules. Going to let you know this is actually going to be a relatively simple one. So I'm really actually excited about this. Here's what you're going to need to know. You're going to need to know about hydrolysis and dehydration synthesis. And they are used to basically cleave and form covalent bonds between monomers. This is all we're doing. This is probably going to be the shortest presentation, which is why I wanted you to do both 1.2 and also 1.3. This one's not nearly going to be as long, so let's get right into it. This is probably one of my favorite pictures ever because what it does is that it actually articulates what you are made of. So if I go ahead and take a look at, Mr. Y right here, hanging out right here, make sure I got the bald head. You get some little glasses right going on in here. Yep, there I am. Cool. And put like my little vest on as well. All right, gang, I am made up of this much water and this much macromolecules. All right. So water, you are primarily, right, they say like you're like 70% water. Yeah, like pretty much most of my body is made up of water. The other like almost like quarter-ish of it, like 18, 19% of my body is made up of these macromolecules. And then there's going to be some ions and small molecules that are in there. There's like your hormones and, well, not your hormones, but like the ions. So like any salts or calcium ions, sodium, potassium ions that are in those sodium, potassium pumps. So you're made of all that stuff. That doesn't matter. We want to focus on this macromolecules right here. And if I go and take a look at my macromolecules, this is how my macromolecules are set up. I am primarily made out of proteins. All right, if I go and take a look at the mirror and I like squish my, poke my squishy face, I'm pretty much saying like, hey, look, I'm made out of proteins. The next most common one is going to be nucleic acids, which makes sense because this is where your DNA is going to be, as well as your RNA. So you are constantly using DNA to create RNA, right, through that process of transcription. You may remember this from your bio, transcription. Okay, we're going to get into this later on. And then its RNA is going to be translated into protein. So this is why these guys are like the biggest out of them all, right? Nucleic acids and proteins, because this is basically what you're doing consistently all the time. In the case of this other stuff, carbohydrates, you're going to be made out of carbs and lipids. All right. So you'll have glycogen that you are stored. You have glucose that is inside of you constantly being used. And then lipids is pretty much just your hormones and your cell membrane. So this trying to get you a snapshot of what you are made out of. It's kind of cool. Right? Do you really need to worry about that? Not really. But what we need to do is talk about how it is that we make these proteins, nucleic acids, carbohydrates, and lipids in the first place. Because I want you to think about this for a second. When you were born, how much did you weigh? Probably like, almost like 8 pounds, so many ounces, 9 pounds, big baby, right? Well, how did you accumulate as much mass over time? Hopefully you answered with saying that you ended up eating, right? Well, the things that you ended up eating, you ate proteins and you eat carbs and you eat fats and stuff. Nucleic acids, right? That's basically your DNA. So anything that was living will have nucleic acids in there. So you basically consume these things and then you're going to go and use them to, they basically make you, right? Hooray! I'm made out of food. That's great. But you have to think about how this actually happens, right? If you end up having a chicken, right? Your chicken right here. Well, it's not like you just like kill the chicken, right? It's not like something happens where we kill the chicken and we end up putting it and we assimilate it to other bodies. No, what happens is that we have to eat the chicken, you cook it, and you end up digesting it, right? And when you're digesting it, you're chemically digesting it in order to turn it into basically building blocks in order for your body to go ahead and use, right? The protein that the chicken is made out of, you can't use it for anything. So you break it down into individual amino acids and then you use it to go and do stuff with, right? We make new proteins out of it. Folks, this is the whole premise of this section, this little lesson right here, okay? So let's go ahead and get rid of this guy right here and move on. Monomers are the units that build larger molecules or polymers. Okay, I think it'd be very ideal for you. It'd be very ideal for you to write down these two terms, monomers and polymers. Okay, basically what happens is that a monomer is an individual unit. So that's a monomer, and that's a monomer. They are individual. They're the individual building blocks. They're like bricks, right? They're like bricks. Well, a polymer is when you start to put them together, right? So now I'm going to start to build them into a bigger chain. And this polymer starts to become a bigger macromolecule, right? Think of it like the bricks, right? What are you able to make with a brick? You get a bunch of bricks. What can you make? Well, you can maybe make a wall, right? You make a brick wall. It'd be the worst brick wall ever. Look how small it is, but I think you get the point, right? We make a brick wall. Maybe I end up taking bricks and using your imagination, I could maybe make like a bridge, right? Make a bridge out of it, right? Maybe something else I can do with bricks. I can make an oven, right? Old timey. I'm not going to go be crazy with this, and here's like a little open space, and I'm not an art teacher, everybody. This is probably the worst brick oven you ever seen, right? There's a brick oven, right? Little smoke coming out there. Look, the person I'm trying to get at is that these monomers, these individual building blocks, will build into polymers. But every polymer can be slightly different, right? You're building them into different things. Kind of like how I can take bricks, right? Bricks, or I can take carbs, or I can take amino acids, and I can build some pretty cool stuff with them. And that's what I want you to be aware of, okay? With that out of the way, the two processes that we need to talk about to make this happen is dehydration synthesis and hydrolysis. We're going to go into more detail about both of them, so don't worry, you don't have to pause the video. We're going to get right into it. But they have to do with water, all right? And we're going to show you how. Condensation reactions. This is your dehydration synthesis, all right? If you are dehydrated, what are you lacking? Hopefully you said water, right? All right, so you end up being dehydrated. You are losing water. You have less water. Dehydration synthesis, right? When you synthesize something, you are making something, right? So H2O, like dehydration, H2O, like loss, building, right? That's kind of what this is saying, right? Dehydration synthesis, H2O loss, building. Dehydration. I'm going to remove water, this guy right here, and I'm going to go build something out of it. This is how we build polymers. That's how we build polymers. All right, this is big. So we end up seeing that we end up having this monomer right over here. We got this monomer right over there. And what we're going to go do is that we are going to end up taking this OH group right here and this H group right there, and we are going to remove them, right? We are literally doing what we're saying right here, right? The H2O loss, right? We are removing the H2O. That's the dehydration synthesis. That's the dehydration portion of this. So I've lost water, okay? And then as we can see, the building, I have now built a polymer. All right, that's the process. Dehydration synthesis, you are removing water in order to go and build a polymer, right? We said building polymers. So what are some kind of polymers that this process uses? Well, this is how you make proteins, right? When you talk about translation, right? When we do that in unit 6, this is going to be like a wow around, right? Translation, right? This is how you end up creating the proteins in like the ribosomes, right? And you this dehydration synthesis, this is how this happens. If you're doing DNA synthesis, right? If you have to go and copy the DNA, we're part of the cell cycle, right? Synthesis. And you have to go and create a new strand of DNA from an old one. Well, you build it using these nucleotides. We'll talk about them later, but that's going to be another way where we're going to use protein synthesis. And then also, maybe in the case you have too much glucose and we have to go and create glycogen, this is how we store glycogen, right? This is how we end up storing glucose that's too much, right? You have insulin, and then it ends up having it absorbed by the cells, or it's going to end up going to the liver and stored as a glycogen in case you're hungry and your blood sugar is low. We release it, right? So these are different condensation reactions. Again, the basics of it is that you're taking these individual parts, we're using an OH group and an H group on another, and then you're just linking them together and you have H2O left over. That is dehydration synthesis. And this whole picture is just showing you can do this over and over and over and over again, and you can make some really, really big polymers. This is how stuff gets built. All right, we lose water and it's being released. Hey, guess what? Now we're going to go into hydrolysis. Hydrolysis. Hydrolysis literally translates into water splitting or water cutting. Water cutting. Remember in dehydration synthesis, we ended up losing water and it was able to build the things into bigger bits? Well, now what we're going to go do is that I am going to go and take my big old polymer and I'm going to go throw water at it. And what the water ends up doing is that because it's H2O, right? It's an O and we have an H here and an H right here. Well, guess what? This this H over here is going to go over to this part. And I'll do this in different colors so you can kind of see like what's happening, right? Might be easier for you to see like in different colors, right? This OH, right? That's going to go over to here. There's the OH right over there. And then the purple H, right? The purple H ends up going to right here. Okay, so that's supposed to show you like, okay, here's my H2O. Boom! I end up using it to split the polymer apart. And now I have my OH on one and my H on the other. It's going to just end up happening, right? The chemical reactions take place. You do not need to know the mechanism why it happens. Just know that dehydration synthesis and hydrolysis, we're basically using water in order to either, create these polymers or break them apart, digest them into smaller pieces, right? Because now let's say that let's say that this was starch, right? You like potatoes? You like starch? Right? I love starchy food. I love pasta. It's delicious. Well, when I end up taking the starch, right? I go out to, Olive Garden. I end up eating my delicious pasta meal. Well, sure enough, I can't just use the starch. I can't just use the pasta as is. I need to chemically digest it. So what's going to happen is I'm going to take some water, right? I drink my water, right? So now I have water in my system. The water is going to be used with some other enzymes, some other stuff is happening, and I'm going to start to break apart the starch into individual glucose molecules. And now this glucose is small enough to go through, my phospholipid bilayer, and it can actually be used by my cells at this point. So chemical digestion is necessary in order to break the big stuff apart, so that way it's a lot more easier to use. All right, you're either digesting or you're building. That's what you're doing. You're either breaking things down or you are building things up. And this last image is just kind of a take-home thing to show you how it all goes, right? We can end up seeing that if we go in this direction, we have a dehydration synthesis, right? Condensation, dehydration synthesis. And if we go in this direction, we have hydrolysis. We are using water to break it apart. Notice it has that OH group and that H group right there. All right, giving you a heads up. These are amino acids right here. And notice that they have this H and this OH group. Works for them. Over here, I got a carbohydrate. I go take a look. Look, it has an OH and an H group right there. Right there. I'm going to go ahead and remove them. I'm going to take them apart. And then from there, I can end up linking this together. Okay, we can see that this oxygen right here, this oxygen right there, is the same oxygen that's right there, and it's holding it all together. Gang, this is what is happening in your body every single second of every single minute of every single hour of every single day, is that you have all these chemical reactions that are breaking molecules down, building them back up in order to maintain homeostasis inside of you. It is a phenomenal process, and it is incredible when you think about all these little tiny biochemical reactions that are taking place and doing the things that they need to go and do. What I want you to think about with this, right, is that we talk about these intermolecular forces and stuff like that, right? We talked about that with water. Think about this, how it's that these molecules, right, have certain behaviors, that this is going to be the place that we want to go and focus and do our hydrolysis. That kind of looks like a skeleton right there. You see that? Like, look at that guy right there. I'm diurous. I'm going to go get rid of that, right? But when you go and take a look, right, maybe it's hard for these enzymes to interact with these areas because the electronegativity is super, super, low over there, right? And what have you, right? Maybe it's that I can't take the H over here, right? The OH over here and the O over there. Chemically speaking, that's not going to go and work. Okay. These biological macromolecules evolved in a way. I want to say that, man, evolving is a really tricky term to go use, but basically evolution use these amino acids to be able to, or use these biomolecules, I should say, in order to progress life how we see it right now. Now, this is going to be me going on to the soapbox and stuff, but it's just kind of fascinating to think about that this is the biochemistry that we know of and what's happening in your bodies and all that other fun jazz. Just at a minute, right? If there was different biological or macromolecules from like some other place, like thinking like alien life or something, it's just something to kind of think about, right? Whereas like, what does that biochemistry look like? And I'm telling you, like, we could go down that rabbit hole, but I digress. That's all you need to know about, folks. You need to know about dehydration synthesis. You need to know about hydrolysis. You need to know that H2O is the culprit. And at the end of the day, are we trying to make a polymer or are we trying to make monomers? What is your goal and how are you going to go and do it? That's it, everybody. That's going to be it for the first 3 sections. So 1-1, 1-2, and 1-3. You are going to go ahead and prepare for your quiz. And I am going to wish you the best of luck. Please make sure you check Schoology for all the details for that. And I will catch you on the next one. See ya!